Welcome, everyone, to one of the most fascinating mysteries in evolutionary biology. Imagine you are an archaeologist who just discovered a strange, ancient vehicle that is half horse-drawn carriage and half automobile. This bizarre hybrid would perfectly show you exactly how human transportation evolved from animals to machines over time. In the animal kingdom, a tiny caterpillar-like creature called the velvet worm, belonging to the phylum Onychophora, serves this exact same purpose. Today, we are going to explore how this incredible organism acts as a living time machine, showing us the exact steps of evolution frozen in time. Now that we have introduced our fascinating subject, let us understand why it is called a living time machine. Why does this matter? Because understanding the transition from simple to complex life is one of biology's greatest challenges. The velvet worm provides a rare, living window into the past. It shows us how ancient, soft-bodied worms might have gradually evolved into the hard-shelled insects and crabs we see today. By studying the phylum Onychophora, we are not just looking at a unique animal. We are literally watching the intermediate steps of evolution that have been preserved over hundreds of millions of years. Building on this idea of a time machine, let us look at how it connects different branches of the tree of life. Formally, the evolutionary significance of Onychophora lies in its historical status as a connecting link between two massive and highly successful animal phyla, Annelida, which includes segmented worms, and Arthropoda, which includes insects and crustaceans. A connecting link is simply an organism that possesses distinct anatomical and physiological characteristics of two completely different taxonomic groups. The velvet worm is a beautiful mosaic, perfectly illustrating the evolutionary transition from worm-like ancestors to complex arthropods by blending primitive and advanced traits. To truly appreciate this connecting link, let us first examine its primitive side. Why do we compare them to earthworms? Because when we look closely at their anatomy, onychophorans exhibit several primitive annelid-like traits. They have a continuous muscular body wall, much like a standard worm. Their legs are simple, unjointed appendages, lacking the complex joints seen in insects. Furthermore, they possess segmentally arranged nephridia for excretion, which is a classic characteristic of segmented worms. These features anchor them firmly to their ancient, soft-bodied ancestors, providing the baseline from which more complex traits eventually evolved. Having seen their primitive traits, let us now look at their advanced features. This is where the evolutionary leap becomes visible. Despite looking like worms, onychophorans possess advanced arthropod-like features. They breathe using a tracheal respiratory system, just like insects. They also have an open circulatory system with a dorsal heart, and their body cavity is a hemocoel. Because of these advanced traits, modern cladistic taxonomy places them in the clade Panarthropoda. This suggests they are a sister group to arthropods rather than a direct ancestor, perfectly showcasing the gradual accumulation of complex biological machinery. Now that we understand their anatomy, we must ask, why does this strange little velvet worm matter so much to science? Understanding Onychophora is crucial because it solves the biological puzzle of how complex, jointed appendages and hard exoskeletons first originated on Earth. Without these creatures, the evolutionary jump from a squishy, segmented worm to a highly armored, flying insect would seem completely abrupt and almost impossible to explain. They provide living proof that evolution happens in gradual, cumulative steps rather than sudden leaps. Furthermore, studying their unique biology, like their sticky slime, inspires real-world biomimicry and new synthetic adhesives. This brings us to an important question. How do we actually prove when these evolutionary changes happened? We cannot just look at fossils because soft-bodied velvet worms rarely fossilize well in the geological record. Instead, evolutionary biologists use a brilliant mathematical concept called the molecular clock. This concept relies on the steady, predictable accumulation of mutations in DNA over time. By comparing the DNA sequences of a modern velvet worm and a modern insect, we can count the genetic differences. This allows us to calculate exactly how long ago they shared a common ancestor, giving us a precise evolutionary timeline. Now that we know what the molecular clock is, let us look at the mathematics behind it. This beautifully simple equation comes from the idea that mutations build up at a constant rate. The formula is D equals 2 times R times T. Here, D represents the total genetic distance, which is the number of differences between the DNA sequences of the two species. The symbol R represents the mutation rate, which is the known rate of genetic changes per million years. Finally, T represents the time in millions of years since they diverged. The factor of 2 is crucial because both lineages have been accumulating mutations independently since they split. If the genetic distance increases while the mutation rate stays constant, the divergence time must be older. Before we move on, let us see this formula in action with a concrete example. Suppose we are comparing a specific gene between a velvet worm and a centipede. We are given a genetic distance of 0.12 and a mutation rate of 0.0001. We need to find the divergence time, T. We start with our formula, D equals 2RT, and substitute our known values. Multiplying the mutation rate by 2 gives us 0.0002. We then divide 0.12 by this combined rate. The result is 600. This physically means that the lineage leading to modern Onychophora and Arthropoda split approximately 600 million years ago, deep in the Precambrian era. Now that we have used this formula, let us understand the algebraic manipulation we just performed. In our previous example, we solved for time by plugging numbers directly into the base equation. However, in evolutionary biology, we often want a direct pathway to solve for the unknown time using our known genetic data. To do this, we start with D equals 2RT and set out to isolate the time variable, T. By dividing both sides of the equation by the term 2R, we cancel it out on the right side. This leaves us with T equals D divided by 2R. This is a crucial first step before plugging in any raw numbers to avoid calculation errors. Continuing from our rearranged formula, let us look at what this actually means physically. The denominator, 2R, represents the combined total rate of mutation for both evolutionary lineages as they diverge over time. By dividing the total genetic distance by this combined rate, we yield our final time. This mathematical relationship is profound. It shows that if the mutation rate stays constant, any increase in the genetic distance D mathematically proves that the divergence time, T, must be much older. This simple fraction is what allows scientists to prove that species with vastly different DNA separated much further back in evolutionary history. This rearranged formula leads us to an important comparative problem. Let us calculate the divergence time between a velvet worm and an earthworm, representing Annelida. We are given a larger genetic distance of 0.18, and we assume the same mutation rate of 0.0001. We use our rearranged formula, T equals D over 2R. Substituting our values, we divide 0.18 by 0.0002. The calculation yields a massive divergence time of 900 million years. This tells us that their last common ancestor lived extremely far back in the ancient history of the Earth, implying a very deep evolutionary split. Now, let us perform the exact same calculation for the other side of the evolutionary tree. We will compare the velvet worm to a fruit fly, representing Arthropoda. Here, we are given a smaller genetic distance of 0.10. Using the exact same formula and mutation rate, we substitute our values. We divide 0.10 by 0.0002. The calculation yields a divergence time of 500 million years. This shows that the evolutionary split between velvet worms and arthropods happened much more recently in geological time, providing a stark numerical contrast to the older annelid split we just calculated. So, we have successfully calculated both divergence times. This brings us to the mathematical proof of their classification. We are setting out to prove mathematically who Onychophora is more closely related to. We have two distinct times. The split with Annelida happened 900 million years ago, while the split with Arthropoda happened 500 million years ago. The logical reasoning here is straightforward. In evolutionary biology, a more recent divergence time always indicates a closer genetic and evolutionary relationship. We will now compare these two values to finalize our proof. Let us conclude our mathematical proof. We compare the two divergence times directly. Because 500 is less than 900, this mathematically proves that Onychophora shares a more recent common ancestor with Arthropoda than with Annelida. Therefore, despite their worm-like appearance, velvet worms are evolutionarily closer to insects and spiders. This mathematical proof beautifully supports the anatomical evidence we observed earlier, perfectly validating their modern classification in the clade Panarthropoda. The numbers and the biology align perfectly. To visually reinforce what we just learned and proven, let us look at this cladogram. A cladogram is a branching diagram that shows evolutionary relationships. On the horizontal axis at the bottom, we have time in millions of years, moving forward as we go right. Notice the first oldest split on the left, which branches off to Annelida, representing our 900 million year calculation. As we move forward in time, we reach a circled node labeled clade Panarthropoda. This represents the more recent 500 million year split, where the lineage divides into Onychophora and Arthropoda. This diagram perfectly maps our mathematical results onto the tree of life. We have covered the full topic, so let us summarize our key takeaways. First, the evolutionary significance of Onychophora cannot be overstated. They serve as a crucial biological bridge between simple segmented worms and complex arthropods. Second, they showcase a fascinating blend of primitive and advanced traits, proving that evolution happens in gradual steps. Finally, through both anatomical observation and mathematical molecular clock calculations, we can confidently place them as a sister group to the arthropods. You have now seen how biology and mathematics combine to illuminate the dark, ancient pathways that led to the incredible diversity of life we see today.